everyone, it's your host, Alyssa. We didn't have a podcast last month, and that's because I was actually facilitating the BioIT Hackathon, which was April 1st and 2nd, on behalf of the NIH Common Fund Data Ecosystem Training Center. This is actually my fifth time organizing a hackathon as part of the BioIT World Conference and Expo in Boston, and it's always a lot of fun. If you're not familiar, this conference brings together a really amazing mix of data scientists, developers, and life science pros, all working together to solve real-world data challenges. So let's talk about my favorite topic, hackathons. These events bring diverse groups of researchers and tech folks and are all about collaboration, innovation, and really a fast-paced problem-solving space. They're hands-on, they're fun, and they're packed with opportunities to learn from your peers, to make open source contributions, and to come up with some really creative solutions. Plus, there's a strong focus on making tools and data more accessible and usable for everyone. This year's hackathon centered specifically around fair data that's findable, accessible, interoperable, and reusable, and we really leaned into open source tools and platforms as well. A big part of the challenge was to improve interoperability using CFDE data and tools. And over 2 packed days, 5 fantastic teams worked with CFDE datasets from Glygen, IDG, HubMap, and MotorPack. And even the one team that didn't focus specifically on CFDE data still found a clever way to weave CFDE tools into their work. I gotta say, the participants this year absolutely blew me away, although to be fair, they do every year. But this year they were incredibly creative, hardworking, and brought such a positive energy to the event, which is why they're always my favorite. It's always inspiring to see how quickly people come together, share ideas, and build things that can make a real difference in such a short amount of time. Before we dive into the presentation, just a heads up, these were originally delivered with slides in video format. So if you want the full experience, check out the video version on either Spotify or Apple Podcasts. All right, let's hear from our amazing teams.
So I have 8 minutes to present this, and then we're also going to have a live demo. So if you have questions, put them in Slack. There's not going to be time because these amazing people did a ton of work here, and I'm very, very proud of this team. It's really amazing. Really, the problem is that we don't treat cancer patients very effectively, right? So we know that ovarian cancer and colorectal cancer and non-small cell lung cancer and bladder cancer all have specific subtypes, but we don't treat people based on the subtypes, and so they go through unfortunate treatment regimens and often die. And it's really unfortunate. So what we would like to look at is not only drugs for subtype specificity, but drugs for driver mutation specificity. We think those combinations will actually give cancer patients the best shot. And so what we want to do is do in silico predictions of that these combinations that can be then tested in cells. So that is exactly what we're doing. I already told you about the different kinds of cancer. And we're leveraging Drug Central to look at safety and efficacy in terms of what these drugs look like. So here's our workflow. We're calling it OncoMatch, so cancer type plus gene mutations. We use Cosmic and a bunch of information from Drug Central to look at available drugs as well as structural similarities from with other drugs, and then we think about activity scores against drug targets. This is what it looks like. Here's the sum of genes per cancer type and also the sum of genes per drug target. And then we're able to plot this, and my colleague Shelby is going to present that.
Yeah, so here we have a couple radar plots. Both of these are for bladder cancer specifically. But yeah, we want the spikes to kind of represent drugs with like a higher efficacy. So on the left we have IC50 represented, which is concentration of a drug needed to inhibit a biological process by 50%. For the purposes of visualization, we took the inverse of that, so that spikes would show a higher efficacy, because we want a lower IC50 to represent higher efficacy. So for Viz, we took the inverse. And it's the same deal with KD, where a lower value shows a higher binding strength. So we took the inverse as well. But here we can see drugs that have like certain spikes, which will likely be more effective in the treatment of these cancers. And then I'm going to hand it off to Stan for a demo where we can see more radar plots and more visualization across different cancer types. Yeah.
All right, Stan, take it away.
Thank you.
Yep.
So, all of those analyses are interactive. So you are able to like do them interactively based on your like choice of the cancer and drugs. Let's go through the workflow. First, you select the type of cancer. The types of cancer are from the Cosmic database, and here we have 4, but it's easily extendable to like multiple different types. Yeah. So, for example, when we choose, let's choose a non-small cell lung cancer. Yes, that's right. That's right. So what it does, it's like it fetches from the Cosmic all of the genes that are related to this type of cancer, and then from the Drug Central, it fetches all of the drugs that have like some experimental data on targeting this gene, and then these are very small, but we can see here that here we have like values for basically the for all of the genes which are related to this NSCLC. We have some drugs which are targeting those genes, and here we have KD and IC50 for all of those drugs. And then here we see like the network of the connections. This is basically very simple visualization, bipartite network. So those are the genes that are related to this type of cancer, which are fetched from the Cosmic, and those are the drugs that are targeted by those genes, that are targeting those genes, which are fetched from the Drug Central. And here we have connections between them. So, yep. And then we can like also further explore those drugs. So, for example, here we have figratinib, let's say. Yeah. So let's select it. Okay, I mispronounced it. So if we select it, it also fetches from the, what's the name of the database?
Drug Central.
No, but the similarities to other, it's like, it's proteomics data from.
So there was a deep coverage proteomic mechanism of action study where we took the experimental data. We mapped the proteomic mechanism of action drug-induced proteomic signatures, and then we calculated the similarity between the drugs based on structure. So if you have one drug that targets a gene for a specific cancer type, then you can potentially find another pair of genes or drug-drug interactions. So we have the structure similarity based on the 3D molecular structure of those compounds, and we also have experimental data between each drug-drug interaction on a proteomic level of how the expression changes as well.
Yep, and you can select multiple drugs from this, like these are that have evidence that they are connected to those genes, and it will find you another potential drugs that have similar proteomics profiles and also similar structure to those. And yeah, that's all. Any questions? All the questions. Yeah. Thank you.
We are Autopath, so our goal was to get to the endpoint of interactive pathways for immune-related diseases. The problem is, how do we leverage existing data to understand disease? And the challenge, too much data, hard to find, could add siloed analysis tools, not very user-friendly for biologists, requiring too much comp bio wrangling. Okay, Autopath, the answer. Interactive pathways for immune-related diseases. Create a workflow to transform single-cell RNA-seq data. In this case, it was actually, for the data we're seeing, single-nucleus RNA-seq data and get to enrichment analysis and ultimately to visualization. Access the data where it lives. So data may be in AWS or GCP or in Japan. So AutoPath flow. We start with AWS data. So actually the data are in cell by gene. We run a tool on a platform as a service called LifeBit NF Copilot on AWS. Thank you, AWS, for credits. We're using a tool called NSForest. That stands for Necessary and Sufficient Markers. And you get marker genes and binary genes for cell clusters. We do enrichment analysis for those top binary genes we focused on, binary genes for a specific cluster. In this case, we're using RPE, which is a retinal pigment epithelial cell, I believe. And we run 2 enrichment analysis, Enricher, which is a Common Fund data ecosystem project, and the new Pigeon we executed today. We take that, we then go to Terra and do gene set enrichment analysis and differential gene expression analysis, and we explore pathways. This is the data set. It's the lead data set on cell by gene. Look it up. There are 90 distinct clusters. It's a huge data set. So we're only looking at one of those 90s, just to give you an idea. This is, like I said, it's a free project. What does NF Copilot do? It is their advanced analytics module. It does the elastic compute orchestration, next flow execution, cost management, and workspace collaboration. What I did today is I Dockerized the new release of NSForest with GitHub Actions, blah, blah, blah. Look for it later. H5AD files, NSForest binary genes. RPE, as I said, you'll note there's a cluster size, an F-score. That is the quality of that cluster. This F-score is a very good thing to have in mind. It's got a positive predictive value of 1, so a great set, and these are the 10 genes. And that is it. So just real quick, I don't know my time. This is on Terra. When you run Enricher, you can generate a Jupyter Lab notebook, and the Jupyter Lab notebook is Avi Mayan's creation, and there are many things you can do. You can do transcription factor enrichment, which we focused on. If you go to the Enricher site, you can do other things. These 3 genes, and actually I think you did all 10 of them, these are transcription factors that are significantly enriched with those 10 binary genes, and these are interesting, and we will talk about them momentarily. So these are out of order, I guess. That's another enrichment, I guess. Oh, wait. Oh, then with pigeon data, oh, actually, I just showed you the pigeon data combination. ITXR, was that for the, I don't forget, hold on. Just a second. I just want to look at the list, I can answer that. No, when we combined pigeon data, so then we took those same 10 genes, went to the Common Fund data ecosystem where there's a bring your own gene data set, and the neuronal factors showed up as enriched, which was very interesting, and our neuronal expert confirmed that, and you'll see more about that later, and I'm handing it off. Boom. You have differential analysis.
Yeah, so just quickly talk about the differential gene expression analysis. So we use the CFT GTExite data for that. It has 523 samples and almost 60,000 genes. So there is this MGS classification to classify advanced stage macular degeneration into 4 levels. The advanced level is 4, the intermediate is 3, early stage is 2, the no eye disease is 1. So what I'm working on is to compare the no eye disease, which is the level 1, to any other one. For now, I'll just do greater than 1 will be the ones that are having the advanced stage macular degeneration compared with gene expression in the no eye disease. So I'm working on that already. It's not in a notebook, it's just our scripts that I'm writing, but the goal is to have it in a notebook format that Peter can use. So,
cool. So this is a notebook I generated. So I worked on the more downstream analysis part of this project. So I took output from Leighton and Ann's data sets, and they identified differentially expressed genes. So for my notebook, I took 10 of these genes, or I guess what's displayed is just 3. They're transcription factors expressed in RPE cells that are identified through single cell sequencing, and I did pathway enrichment analysis, so I tried to identify pathways in which these genes were statistically overrepresented. And so I used this Python library called GCPy to do the enrichment analysis. It identified these 4 pathways. My notebook processed this and generated these URLs that lead to data visualizations that Eric will explain. So that's cool.
It's a demo, so let's do it live. All right, so you see this URL, opening it up in a new tab. This is just basically a static HTML page with some JavaScript. It's going out to the Wikipathways database and querying it by this pathway ID. And then you can just pull in basically an SVG file. SVG is really nice because it's programmatically manipulable. And so you saw that one of the genes that's overexpressed is this OTX2 gene that's involved in things like dopaminergic neurogenesis and also, let's see, this other one. Takes a moment. There we are. And endoderm differentiation. And I'd mentioned that OTX2 is a transcription factor for other genes that are relevant for markers for this retinal pigmental epithelial cell. And if you hover over this gene that's colored in here, you can see that it's indeed OTX2. If you want a little bit more detail on like which genes is this interacting with, you can zoom in and see that it interacts, OTX2 interacts with LHX1 and other genes. And this will give you sort of cellular context on the data that you're looking at. You can see that it interacts, in the nucleus rather than the cytosol, not the membrane, et cetera. That's it for the visualization portion. And that's it for our presentation.
Well, I'm the writer.
Oh.
Yeah,
The README isn't done, but it will be great.
Yeah. Thank you, writer.
How to start timer?
I got it. Just go.
Hello. Okay, so let's begin. Hello everyone, we're Team 3. Our project title is Mapping Kidney Disease with HubMap. And firstly, just a brief recap of what we're doing. So in traditional clinic diagnosis of kidney diseases, the physicians needs to look at the histological samples manually under microscope. This is a quite slow procedure, and the predictions of whether the patient has disease suffer from individual experience, insufficiency of experience, so the accuracy of prediction will suffer as well. And also, for different physicians, they have different opinions, even on the same sample, of whether the patient is having kidney disease. So in order to address those 3 limitations, we used 51 healthy tissues from HubMap and that many of disease tissue from KPMP. We did FTU segmentations on those histological samples and extracted features to analyze if there are morphological differences between diseased histological samples and healthy kidney samples. So our project has 2 aims specifically. So the first one is to use machine learning algorithms to quantify the difference between diseased kidney tissue against the healthy reference tissue. So it's basically like a binary classification of healthy or not. And the second aim is to differentiate the disease subtypes into CKD and AKI, because the treatments for these 2 subtypes of disease is different, and it is important to differentiate them in clinics.
This is just a brief overview of the diagram of kind of the workflow in terms of going from the raw histological section to the actual segmentations themselves, and then going through the quantifications, and then eventually the classifications. And so we're using all publicly available data here. The one on the left is from the HubMap website, and the one on the right is from the KPMP website, both using similar visualizations.
So as Yang talked about, the first aim is to differentiate between the healthy tissue and the diseased tissue. So he didn't go over what the diseased tissue that we have. So there are two types of diseases that we have. One is CKD, which is chronic kidney disease. This one is very severe, and in most severe cases, you would need a transplant. And then another one is AKI, which is reversible. If detected early, it's called acute kidney injury. So that is why it's very important to determine between these two subtypes, and that's what part of our aim 2 is. So in the first one, we observed that there are two main functional tissue units that are relevant to this project. One is called the glomerulus, which is basically the filtration unit of your kidney. It is what gets rid of the waste, gets rid of the urine and stuff. And then, so if your kidney is not functioning, the sclerosis, the number of sclerosed glomerulus is going to be more than the functioning ones. So that is one of the predictors that we theorized in this project. And the second one is the tubules, which gets the blood in and out of your kidney. So if your kidney is damaged, there's going to be fibrosis. So essentially, there's going to be gap between the tubules. So as you can see, we calculated the ratio between the damaged glomerulus and the healthy glomerulus as one of the indicators. And for the other one, we calculated the average tubule distances between the centroids of the tubules. We wanted to also calculate the tubule fibrosis, like the amount, but due to time constraints, we were not able to do it. And my friend Kyo will talk about the results of this aim.
Yes. Yeah. Before I start, I'd like to ask for your understanding, as in my English dictionary might not be perfect. I will do my best to deliver the presentation clearly. Today, first, we analyzed 51 healthy kidney and 55 diseased kidney. We compare the ratio of non-clonotic to clonotic glomeruli using a t-test and calculated the 95% confidence interval. The results show that the mean ratio in the disease group was 6.35, while the mean ratio in the health group was 20. As you can see in the graph, the disease group exhibited significantly higher ratio of SG compared to the health group. Statistically, the p-value is less than 0.001, indicating a highly significant difference. This suggests that GS is more severe in diseased kidney samples, and tubular fibrosis is also observed. This study highlights the importance of global GS in kidney disease. We hope that further research will contribute to elucidating the pathologies of kidney disease and developing effective treatment. Thank you.
Oh, and here is just a validation for aim 1. As you can see on the top left graph, the ratios of healthy and disease samples are quite different. We even can draw a line to physically separate them. And on the top right graph, I plotted the ratio of non-sclerosis, which is functional glomeruli, over sclerosis meruli with respect to the tubule lumen fraction. As you can see, we can also physically separate the healthy and diseased group quite easily, even without like running some algorithms like SVM. We can just manually draw a line. And the bottom two graphs is just me exploring if the ratio has the correlation with the lumen fraction, but it appears that there's not.
And this is just another graph of the luminal fraction comparison between the different disease states.
So far, my teammates talked about how we were able to quantify the difference between the functional tissue units in a diseased versus healthy slide. I'll be talking about how we were able to classify a slide on saying if it's diseased or healthy. So the aim of this project is to be able to classify the kidney diseases, namely CKD and AKI, to be able to look at a slide and say if it has CKD, AKI, or if it's a healthy slide. So this is the data set that we are working with. We had 191 slides, 51 of them were healthy, and 1 or 2 of them have a CKD, and 38 has AKD. So this is a multi-class segment, a multi-class classification. So this falls under weakly supervised learning because we only have slide level labels. We don't have patch level labels. So because of this, we can't go about using a traditional patch level CNN training. So this requires a multi-instance learning based model so that it can just look at a slide and take all the patches as instances and look at each slide. The model learns which patches in the slide show the specific label. So it tries to learn which patches are affecting the slide to be classified into a specific class. So we used a model called CLAM. It is designed for weakly supervised whole slide image classification. So these are we ran it for 2 folds. On the first fold, we were able to predict all 19 slides in their respective categories without any issues. In the second fold, we were able to predict all the healthy slides, all the slides in CKD, but in the AKI, we were mislabeling 3 of them as CKD. So we had an average accuracy of 92% for 2 folds.
Just future directions, we want to add a couple more features to explore and then generate heat maps for this CLAM model to check out which areas of the slide that are predictive of those. So thank you.
So, hi, I'm Mihir, and we have our team, Matt, Maitri, and Xiao. We're going to be talking about how we tried to explore MotorPack data using through the lens of machine learning. So you might remember yesterday we asked ourselves, we asked you, is MotorPack data in its current format suitable for machine learning analysis? And we started off, yes. And we did some more exploratory data analysis, and we were like, yes. And did some more, and that answer changed to maybe. And yesterday, at the end of the day, we were like, probably not. So we tried our best. We tried to start, looking at the data, and I'll kind of talk about some of the problems that we ran into. But we started looking at, as you'll remember yesterday, we talked about we had a shotgun approach. We were going to explore 3 different projects and see, is this going to be effective? Are any of these approaches going to work out? And so we had our 3 projects were tissue-based transfer learning, physiological outcome prediction, and sex-specific feature classification, or I guess more. It was kind of also intertwined with like temporal trajectory modeling. And the drawbacks were that we do have a large number of participants in this study, or at least the rat phase of the study. Our N is about 300, but when you consider what assays were run on which tissues, integrating this entire data set was, like our N started to become around 4 or 5, which, is not particularly great for applying machine learning to a data set. That's still a small, even if you have, 10, 20,000 features actually more like 100,000 features, but even if you have that many features, when your sample size is low, you're not going to get anything meaningful out of this. And for temporal modeling, that was also a kind of a relatively small number of time points, and you can imagine that things aren't going to get super effective. So we settled on kind of on the final thing, which was given this low number of points, can we just predict a single thing? Can we just predict physiological outcomes? So the two, kind of lowest hanging fruit are VO2 max and weight. And so those are the two kind of target variables that we're going to look at and see if we can predict based on genomic or omic profiles. And so we wanted to, the way that we approached this was leveraging the existing R data packages that MotorPack provides, and we wanted to identify also like the data sets of interest, like is data available across all these tissues, across all these assays, like which data sets make the most sense to use. And what we landed on was utilizing the transcriptomics and epigenomics data sets that MotorPack provides. Some of the data set, or the data sets were pre-normalized within each assay by MotorPack, and there was a feature to gene mapping. So you can imagine chromatin unfolding, like there's multiple spots where chromatin will unfold, and that might map to multiple genomes. So what we did was we mean normalized, or we took the mean across like the same gene, or the feature to gene, if that makes sense. So there might be like 8 metabolites that map to 2 genes, and then we mean like 4 for each one. And so now I'm going to hand it off, and we're going to talk about the models themselves.
Yeah, so Mahir makes it sound simple. It was a lot of, I think he showed up and he was like, oh, this is what my customers need, and he reacted on the fly and got us a really great data set to play with today. So when I think about exercise, I think about losing weight. So that's where we went as a simple physiological outcome to look at. And this, in the interest of fair, is a plot that MotorPack provides from their study. And what you can see is that each arrow is a rat, and it's the starting weight and the ending weight at the end of the trial. And so you can see that the rats that were exercised for 8 weeks, they lose a lot of weight, and 4 weeks, they lose a little weight. And the sedentary ones, these rats actually sat around for 8 weeks. So there's kind of this intuition that you need not just total weight loss, but weight loss over time. So that's actually the physiological parameter we went after. And we controlled for groups the various weeks, or if it was the control group, and also sex. So when we split the training set, we tried to make sure that not everything was a control in the training set, and we used scikit-learn for that. We applied a lasso model. I'd done lasso before. This is actually lasso Lars, which is apparently better for huge feature sets. There's one hyperparameter, alpha, so we did a little hyperparameter tuning. Basically, if you let the hyperparameter get too large, it collapses all your coefficients to 0, and you end up with a constant MSE. But down here, we found a good one, and we have a little bit of a model. I believe this outlier is killing me, but the rest of it I'm pretty happy with. And then the rest of the team did some bioinformatics magic, and we took our features, we got 8 of them, and got genes, and apparently these, I don't know any bioinformatics, but that identifies pathways in lipid metabolism, autophagy, and cytokine. So the answer is yes, you can do machine learning. You just need a lot of work. And then Maitri tried a different model.
So I tried predicting VO2 max. which is basically how much oxygen you can take up and utilize during an intense activity period. And my answer would be no, using this predictor, using this target variable. So I used, yes, just some data pre-processing before getting into the model. And I used something called gradient boosting regression, which is an ensemble model. So it uses multiple weak model predictions to give us a final model. It's supposed to be, this is the first time I used it, and it's supposed to be better for like highly dimensional multi-omics data set that we have, also kind of prevent overfitting and stuff like that. So this model did not, I mean, I didn't get any positive results. The R square was 0.23. We also did feature selection, so after that the model R square was 0.23, and the MSE was huge, very, very large. So I think I was trying to interpret the results, and initially the plot that Matt showed from the MotorPack data analysis, I should have put the plot here for VO2 max, but that showed very random kind of fluctuations for males and females. So the larger VO2 max you have, the better kind of endurance ability you have. And we saw a lot of fluctuations going down and then up, and that the amount it went up also differed between individuals. So I'm trying to think about why this didn't work, and maybe there are a lot of other factors that are responsible for VO2 max prediction that we probably couldn't account for in this data set. Yeah, and so our major takeaways, so I have only used a single ohm data before for machine learning prediction, so it was really nice to look at how we can use multi-omics data, and also I got to try a new model, which was pretty cool as well.
Yeah, I'll jump back in. As an ML practitioner, I knew just enough to be dangerous. I should have spent more time reading MotorPack's website because they had done a lot of work, but maybe they could have written it more for ML considerations. And we struggled a lot with the data set. At Regeneron, we have something called the data catalog, and I think that would be really handy in this use case.
Thank you. So we would, yeah, so, you know, as the MotorPack representative, we just, what we're going to do is take, you know, what we're going to take away from this is we need better documentation, especially for non-R users, more integrated data structures, so we're not like spending the entire first day just like looking for what data to use or trying to figure that out, and do some more like data warehousing. And so that's everything we have for you today.
So first of all, about the project Glycoenzyme Expression Atlas, linking differential expression to pathway dysregulation. So why this project? So despite the critical role of glycosylation, which is essential in health and diseases, it influences various cellular and biological pathways. It is still underexplored. And glycoenzymes expression patterns and their impacts on the biological pathways are poorly understood. So because of which there is limitations in our current resources, and the existing databases fail to link differential glycoenzyme expression with pathway dysregulation across diseases. And so we have got research challenges. There's difficulties in uncovering diseases mechanisms and identifying therapeutic targets related to glycosylation due to these gaps. So moving on.
Okay, so I'm going to be talking about the methodology of this project. It was a really fun hackathon because we started to build this on the go. Shout out to our team lead, Vlado, and to Julie for helping us. And shout out again to Vlado for helping us navigate the glycogen database, because we started off with integrating glycosyl transferase and glycohydrolase data sets and the gene list. We integrated these two gene lists from the glycogen database, and after integrating them, we found 906 glycogenes
from which we found that most of the diseases associations with glycosylation, the disease that associates most with glycosylation with the set gene list that we selected was muscular dystrophy, as you can see from these graphs. So using muscular dystrophy as our target disease to pursue the analysis, we selected the RNA-seq database from Gene Expression Omnibus database, and after selecting two such data sets, we performed differential expression analysis using DE-seq2 and EDGE-R. And after finding, thousands of differentially expressed genes, we filtered out the glycogenes from our set gene list. And after comparing this differentially expressed genes, we saw the genes that were upregulated and downregulated, and we further went on to perform pathway analysis using KEGG. And after pathway analysis, we also performed network analysis using Cytoscape. And all of these results were validated and consolidated and put into our Shiny app. So we developed the our Shiny app as to further show that we can use the same analysis pipeline for other diseases using the same database. So like we found glycogene and we performed analysis for muscular dystrophy, there's a way using the our Shiny app we can do it for various other diseases like cancer, neurodegenerative disorders. So Isha's going to be talking about the results.
Yeah, so for this particular disease, we were able to find around 800 genes that were differentially expressed. After performing the differential expression, we also tried to find out the common genes between both the data sets, some of the genes that were upregulated and some that were downregulated. So after after finding out the common genes in both the data set, we tried to validate it using Pigeon, and we were able to find that muscular dystrophy is a part of the genes that we shared with the database. And as you can see, the last one out here. There were other human traits that shared the similar genes, and these genes can be further explored with the same pipeline that we have developed. We also used Cytoscape. So basically, these three pathways were altered in these particular patients. Yeah, so just we'll show you guys the our Shiny app and then talk more about it.
So this is the our Shiny app that we developed. So the methodology is present here, and you can see we've used the same pipeline, so it can be used for other diseases. So we showcase the number of genes that were found, the number of glycogenes that were selected, and the significant differentially expressed genes. And I've also showcased the most upregulated gene and their log fold change value and the downregulated gene. So here you can see for the disease that we selected, muscular dystrophy, I have shown the summary of the experiments from the gene expression omnibus and the sample information and the clinical characteristics of the particular disease. I've also mentioned the list of the so this list basically shows all the glycogenes that was used for cross-comparing with the differentially expressed genes. I also have showcased the volcano plots and the other the standard plots that you see with doing differential expression analysis, like heat maps and volcano plots. So this can be modified for any gene. So if you use a GSE ID from the gene expression omnibus, you can perform the same analysis for various diseases, and then also the results for the pathway analysis. Because of time constraints, I was not able to plot or showcase more visuals, but hopefully by the end of the documentation, the our Shiny app is more concrete. So thank you so much, guys.
Thanks so much for tuning in. I hope you got a sense of how energizing and fun hackathons can be, and how powerful CFDE data is when it comes to driving discovery and collaboration. We've got more exciting opportunities to get involved coming soon, so if you're interested in joining a future event or learning more, feel free to reach out. You can always contact us via email. We'd love to hear from you. Until next time.